We continue our lecture with non-specific innate immunity, means the second line of our immune system. So as you see, the second line, same as the first line of defense, is non-specific type of immunity. And on today's lecture, we're going to cover 5 parts of non-specific innate immunity. Those are phagocytosis, the complement system, inflammation reaction, the interference, and natural killer cells. We start with phagocytosis. Phagocytosis means engulfment of one cell by another. What kind of white blood cells play a role of phagocytes? Those are macrophages and neutrophils. Macrophages start as monocytes in the bloodstream, and there are long-lived types of phagocytes. They live in the body of humans up to several months. Neutrophils is the second type of phagocyte, and this is the most numerous white blood cells. Those are the most numerous white blood cells, and they are short-lived phagocytes. When they're not activated normally, they live in our system for about 5 days. When they get activated in tissues, they survive for about 1 or 2 days maximum. So basically what phagocytes do is, the first step, they recognize pathogens, then they attach to their surfaces. The next step, they will engulf pathogens, and then they destroy them, and finally they will expel debris into the bloodstream. So the first step of phagocytosis is recognition. Because phagocytosis is a part of the second line of defense, innate immunity, and you remember it is non-specific line of defense. It means phagocytes will recognize common molecules on the surface of the pathogens. For example, peptidoglycan on the surface of gram-positives, or LPS on the surface of gram-negatives. So how do phagocytes recognize those common molecules on the surface of the pathogens? They use specific structures that we call opsonins. Different structures can actually play a role of opsonins. For example, immunoglobulins or antibodies sometimes play a role of opsonins. On this slide, they give you as an example C3b complement component. So the components of complement system also sometimes play a role of opsonins. So what is it, opsonins? That is a structure that phagocytes use to recognize common molecules on the surface of the pathogens. So basically, they play a role of bridge between phagocytes and pathogens. And if you look at this picture on this slide, you'll see this is our opsonin. This is a phagocyte. The orange one is a phagocyte, and the green one this is some kind of bacterial cell. So as you see, what opsonins do, they recognize certain structures on the surface of the pathogen and attach to that surface, and then phagocytes will attach to the surface of opsonins. When phagocytes attach to the surface of the pathogens, this step of phagocytosis is called adherence or attachment. Alright, so on this next very nice slide, you see the pictures that cover all 4 stages of phagocytosis. The first step is called recognition, and you see some kind of bacteria, opsonin, and phagocyte. It already recognized that pathogen and attached to the surface of that pathogen through the opsonin. The second stage, once attached to a bacterial cell, our phagocyte will extend its pseudopods and will engulf that pathogen. This step of phagocytosis is called engulfment. The next step is called destruction of engulfed pathogen. As soon as pathogen gets inside of the phagocyte, it will be covered with vacuole. And then phagocytes will release enzymes. Specific enzymes will destroy pathogen within phagocyte. This is what we call destruction. And finally, the last step of phagocytosis is called expulsion. Expulsion means debris of destroyed pathogen are released into the bloodstream. So the whole process of phagocytosis takes about 30 minutes or even less to destroy engulfed pathogen. What you have to remember is the debris of the pathogen will be released into the bloodstream. Remember, the last stage of phagocytosis is called expulsion. For our immune system, for the third line of defense, those debris of the destroyed pathogen will play a role of antigens. And that means our immune system, the third line of our immune system, will start producing antibodies against those antigens. So we can say that phagocytosis actually can stimulate our third line of defense. The phagocytosis is extremely effective in destroying most pathogens, but some bacteria can survive phagocytosis. As an example, we can use Mycobacterium tuberculosis. Because it has waxy cell wall in its structure, Mycobacterium tuberculosis can survive within phagocytes, and even it can multiply within phagocytes. So that means phagocytosis is not effective against Mycobacterium tuberculosis. The next part of the second line of defense or innate immunity will be complement system. Complement is a set of microbe attacking proteins that can be found in the bloodstream, lymphatic circulation, and extracellular fluids. In order for the complement system to start working, it has to be activated. When activated, complement system plays several important roles in both the innate and adaptive immune responses. And you have to remember that innate immunity and adaptive immunity actually activate complement system in different way. Activation of complement in the innate response proceeds through what we call the alternative pathway. The alternative pathway means activation of complement system in absence of antibodies. And that's clear why, right? Because innate immunity doesn't make antibodies. Antibodies are part of the third line of defense or adaptive immunity. So that's why innate immunity activates a complement system in absence of antibodies. You don't have to know the details, great details of alternative pathway of activation of complement system, but we're going to stop on main points. So everything starts with C3 complement component. The first step, that complement component within bloodstream will be hydrolyzed. It means it will react with the water. After it's been hydrolyzed, it reacts with factor B and factor D from the bloodstream. And then there will be a cascade of biochemical reactions, and the end products of activation of complement system will be C3b and C3a complement components. Let's talk about function of C3b complement component. You already know that C3b complement component can play a role of opsonin. So we can say C3b complement component activates phagocytosis. Also, C3b complement component is a part of what we call membrane attack complexes. Membrane attack complexes are part of complement system, and their main function is to destroy microorganisms, pathogens. So how do they do that? They recognize surface receptors, surface molecules on the pathogens, sit on top of those surface molecules, and then start making holes within cell membranes of those pathogens, and this way they destroy it. So once again, C3b complement component is a part of membrane attack complexes. And finally, C3b participates in producing of C5a complement component. And the role of C5a is to attract phagocytes to the areas or site of infection. And the next part of the second line of defense is an inflammation. The inflammation is non-specific defense response by the body to trauma. As you see on this slide, inflammatory reaction can be caused by injury, infection, or by hypersensitivity reactions or disordered immune response. You have to know the 4 symptoms of inflammatory reaction. Those are redness, swelling, pain, and heat. Alright, let's talk about what kind of factors or substrates will cause all those 4 symptoms of inflammation. Soon after bacteria enter our tissues and begin to multiply, macrophages wait in there to recognize them as invaders or pathogens. Then, with the help of opsonins, let's say C3b complement components, macrophages phagocytize them. As they degrade these bacteria, cellular components are released, which stimulate the macrophage to release compounds or substrates that are called cytokines or inflammatory mediators. So that means all those 4 symptoms of inflammation, redness, swelling, pain, and heat, will be caused by inflammatory mediators. For example, why do we develop redness as a symptom of inflammation? Because inflammatory mediators will dilate blood vessels and will increase blood flow to the site of infection. Why do we develop swelling as a symptom of inflammation? Because inflammatory mediators will increase permeability of capillaries at the site of infection. Why do we develop pain as a symptom of inflammatory reaction? Because inflammatory mediators stimulate or irritate nerve endings at the site of infection. And finally, heat, the last symptom of inflammation. We develop heat as a symptom of inflammation because, once again, inflammatory mediators will dilate blood vessels and increase blood flow at the site of infection. But besides cytokines, what else can play a role of inflammatory mediators? Bacterial byproducts. Remember, the last stage of phagocytosis is called expulsion, when debris of the destroyed pathogen will be released into the bloodstream. They also can play a role of inflammatory mediators. Then the complement components also can cause vasodilation and also can activate white blood cells, means they also can play a role of inflammatory mediators. Next, substrates. Kinins, another example of inflammatory mediators. They also can change permeability of the capillaries at the site of infection. They can cause vasodilation. They can stimulate or irritate nerve endings and causing pain as a symptom of inflammation. On the right side of this slide, guys, there are three different inflammatory mediators that are produced by our own mast cells. And guys, all three of them participate in allergic reactions or hypersensitivity reactions. And the names of those inflammatory mediators you have to memorize and you have to know for your future exams. So those are histamine, prostaglandins, and leukotrienes. From this slide, we're going to stop on this slide. We're going to stop on certain terms that you have to know that are related to inflammatory reaction. The first term is erythema. Erythema means redness. You remember that this is one of the symptoms of inflammation, and we develop erythema as a symptom of inflammation because inflammatory mediators will increase will dilate blood vessels at the site of inflammation and increase blood flow. The next term is margination. Margination is ability of phagocytes to adhere or attach to the capillary walls at the site of infection. And finally, the last term you have to remember is diapedesis. Diapedesis is ability of phagocytes to squeeze through the capillary walls at the site of infection. All right, let's look at the stages of acute inflammation, which is result of trauma. So when certain tissues get damaged, what is going to happen next? Inflammatory mediators will be released. They will stimulate capillaries, the nerve endings. They will activate phagocytes and phagocytosis. So all of these leads to pus, right? Pus will be formed as a result of bacterial infection in this area. Please remember that only bacterial infections produce or form pus. What is it, pus? It's a mixture of dead microorganisms, white blood cells, and damaged tissues. What is going to happen next? Cleanup. Neutrophils and macrophages, phagocytes, will come over to the damaged area and will remove microorganisms, damaged tissues from these damaged areas. And the next stage is called repair. So basically, if damage is not very extensive, tissues will return to normal because a lot of tissues can regenerate. But you have to remember, if damage is extensive, then tissues most of the times are not able to get back to normal, but instead scars will be formed, and that is the connective tissue. That means normal tissue will be replaced with connective tissues in order to fix that extensive damage. But also, you probably remember from your human anatomy class that certain tissues cannot regenerate. For example, nervous system. It means if your patient has brain damage, that is going to be permanent. And finally, the last two parts of innate immunity are interferons and the natural killer cells, or NK cells. You have to remember that those two parts of second line of defense protect us against viral infections. Let's start with interferons. Interferons, those are a group of small proteins that interfere with the viral replication. Let's look at this picture here in this slide. So what you can see on this first picture of this slide is this is some kind of cell. We will call it a host cell, and those are viruses that are going to infect this host cell. As soon as this host cell get infected, it will start producing interferons, small proteins that will interfere with replication of those viruses. Interferons, after they've been produced, will be released by that host cell, infected host cell, into extracellular space. And then neighboring cell is not infected cell will pick up those interferons. After interferons are picked up by an infected cell, interferons will stimulate that cell to start producing AVPs, or antiviral proteins. So basically, if this cell will get infected by the same virus, it's already produced antiviral proteins and they will interfere with viral protein synthesis. So basically, that means that cell will not be converted into pro-virus. That means the replication of that particular virus will be stopped over here. The natural killer cells, or NK cells, this is another part of the second line of defense that protects us against viral infections. What you have to remember about natural killers is, first of all, those are lymphocytes, kind of lymphocytes. Because natural killers are part of the second line of defense, and you remember the second line of defense is non-specific line, that means natural killers are non-specific lymphocytes. And the last one, targets. What are the targets for natural killers? There are three main targets for natural killers. Those are virus-infected cells, cancerous cells or altered cells, and transplanted cells. Let's talk about mechanism of action of natural killers. What you have to remember is each cell in our body has certain receptors on the surface. We call them MHC class 1 proteins. When the cells are healthy, MHC class 1 proteins look in certain way. For example, in this picture, they look purple. NK, or natural killer cells, have specific receptors that they use to check MHC class 1 proteins on each cell. So basically what they do, they go from cell to cell in our tissues in our body, and using those specific receptors, they check the condition of MHC class 1 proteins. What is going to happen if, let's say, our cell got infected with the virus? Then the structure of MHC class 1 proteins on the surface of that cell will be changed. That would be recognized by natural killers, and as soon as they recognize those changes on the surface of the cell, they will produce what we call perforins. Perforins, those are substrates that actually make holes in the cell membrane of our cells, damaged cells, and this is what you see on this picture. So as I said, natural killers, if they recognize changes on MHC class 1 proteins on the surface of the cell, they will secrete perforins, and this way they will destroy that abnormal or infected cell. But please remember, because NK cells or natural killer cells are non-specific lymphocytes, they cannot specifically recognize what happened to that cell, why MHC class 1 proteins got changed, if it's been infected, or it became cancerous cell, or it is a transplanted cell. They just can recognize changes on MHC class 1 proteins and will destroy that cell.